Welcome to today's Explainer. We're jumping right into what is essentially a war, both on a microscopic and macroscopic level, between our domestic animals and a seriously, notoriously resilient pathogen, Salmonella. We're going to unpack exactly how this biological threat operates. And trust me, it's as fascinating as it is terrifying. Let's take a quick look at our roadmap for today. We've got 6 parts. First, the world of Salmonella. Then, how Salmonella attacks cells, followed by the clinical impact on animals. After that, we'll hit stress and disease activation, diagnosing and treating infection, and finally, wrap up with effective control and prevention.
All right, let's kick things off with part 1, the world of Salmonella.
So, over 2,400. That is a massive number, and it represents all the different serotypes within the Salmonella genus. Now, normally, sorting these out is all about identifying specific somatic and flagellar antigens. But the big takeaway for us today, the overwhelming majority of the ones that actually matter in veterinary science fall under just one specific classification, Salmonella enterica, subspecies enterica. It's a bit of a mouthful, but it's the main culprit we're dealing with.
And wow, is this specific subspecies tough? It's mainly transmitted when an animal ingests feces, but here's the kicker. It doesn't just vanish when an animal leaves the pasture. It can survive in damp, shaded soil for up to 9 months. Think about that. It can literally just lie in wait in your farm's soil, in the water, or in animal feed for the better part of a year, acting as this constant, hidden environmental reservoir.
Moving right along to Section 2, how Salmonella attacks cells. Let's zoom in.
Okay, so let's look at the bacteria's sophisticated burglary technique. Step 1 is fimbrial adherence. It attaches itself to the surface of intestinal mucosal cells. Once it's locked on, step 2. It actively forces the host's cell membrane to ruffle. This ruffling is wild because it basically tricks the cell into swallowing the bacteria whole into these membrane-bound vesicles. That brings us to steps 3 and 4. Safe inside this little bubble and using its specific virulence plasmids, the bacteria replicates like crazy and then breaks out. Often, it leaves the host cell with just mild or transient damage as it moves on to wreak more havoc.
So you might be wondering, how does it survive the body's immune system once it's inside? Well, the secret weapon is lipopolysaccharide, or LPS. It's a component of the bacterial cell wall, and it's basically a dual-purpose tool. First, it's a defensive shield. Its long chains stop the host's complement membrane attack complex from destroying the bacteria. But second, it's an offensive, toxic weapon. This LPS is directly responsible for endotoxic effects, meaning it fuels the local inflammation that damages intestinal cells and causes diarrhea. And it's the exact same mechanism that triggers fatal endotoxic shock in systemic infections. Which brings us to Section 3, clinical impact on animals. What does this look like on the outside?
All that cellular damage essentially results in 2 very different types of battles. On one hand, you have enteric salmonellosis. This severely ravages the gut. We're talking fever, depression, anorexia, and this profuse, foul-smelling diarrhea that often contains blood and mucus, leading to severe dehydration. But on the other hand, you have septicemic salmonellosis, and this one is a rapid systemic shock. It hits fast with a sudden onset of high fever and recumbency, which means the animal just can't even stand up. It is incredibly deadly, often fatal within 48 hours if left untreated, especially for highly susceptible young animals like calves, neonatal foals, and pigs under 4 months old.
What's really fascinating here is how incredibly host-specific these consequences can be. The exact same bacterial genus causes wildly different symptoms depending on the animal. Take Salmonella Dublin, for instance. In cattle, it causes enterocolitis, abortion, and even terminal dry gangrene of the hind limbs and ears because of local blood flow loss. Wild, right? Then look at Salmonella Choleraesuis in pigs. It causes enterocolitis and septicemia, but visually, you get this characteristic bluish discoloration of the ears and snout. In chicks and poults, Salmonella Pullorum leads to bacillary white diarrhea and really high mortality, while in adult birds, Salmonella Gallinarum causes fowl typhoid, sudden death, and leaves them with an enlarged, friable, bile-stained liver. Next up, Section 4, stress and disease activation. You might be surprised to learn that a lot of times, this bacteria just sits there quietly without causing any problems at all. It localizes in the mesenteric lymph nodes or hides out as a latent infection in the gallbladder. The critical triggers? Environmental and physiological stressors. We're talking transportation, overcrowding,
pregnancy, or fighting off other infections. Even extreme ambient temperatures, water deprivation, sudden changes in their rations, oral antimicrobial therapy, or surgical procedures. When an animal hits that tipping point of stress, their susceptibility changes. It activates that latent infection, and suddenly they're shedding the bacteria in their feces and getting visibly rapidly sick.
That takes us to Section 5, diagnosing and treating infection. So how do veterinary scientists act as detectives to confirm an outbreak? It's a very methodical, multi-day laboratory process. At 0 hours, you're collecting feces, tissues, or aborted abomasal contents. But because just a small number of bacteria might only mean the animal is a carrier, you have to enrich the sample for 24 hours in specific broths, like selenite F, Rappaport, or tetrathionate. By 48 hours, they do aerobic incubation at 37 degrees Celsius on selective augers. They're looking for visual clues, like red, alkaline colonies with black centers on XLD auger. And it goes on. At 72 hours and beyond, they're doing biochemical profiling, slide agglutination, and phage typing to track dangerous strains, like Salmonella Typhimurium DT104, which is famous for being resistant to multiple antibiotics. And that antibiotic resistance is exactly why treatment is so tricky. You absolutely cannot just guess which antibiotic to use. Because Salmonella frequently carries multiple resistance R plasmids, blind treatment can completely fail. Treatment really hinges on two main things: fluid and electrolyte replacement, and extremely careful antibiotic therapy based on susceptibility testing. For enteric cases, giving oral antibiotics can actually mess up the normal intestinal flora and make the animal excrete the bacteria for even longer. But in those severe septicemic cases we talked about, intravenous antibiotics and immediate fluid replacement to fight dehydration and shock are absolutely critical. Let's bring it home with Section 6, effective control and prevention. So, how do we build a biosecurity shield and keep this out of our herds and flocks? It starts with strict exclusion. Ideally, you want to implement a closed herd policy. If you have to bring new animals in, isolate them until they yield 3 consecutive negative samples, period. When it comes to cleaning, use hardcore disinfectants like 3% sodium hypochlorite, iodophores, or formaldehyde fumigation. Another huge point: avoid using antimicrobial drugs just for growth promotion. That just fuels resistance. And finally, utilize modified live vaccines. They are fantastic for enhancing both humoral and cell-mediated immunity across the whole herd. Now, we also have to remember that this isn't just an animal agriculture issue. It is a massive public health concern. Certain serotypes bridge the gap right into our own kitchens. Take Salmonella enteritidis. This bacteria can actually infect the ovaries of completely healthy-looking hens. This means the organism is transmitted completely silently, directly into the eggs themselves. And if those eggs are undercooked, it leads straight to severe human food poisoning. I'm going to leave you with this provocative thought to chew on. We've just seen how stress activates this disease, how drug-resistant strains evolve, and how easily this pathogen can jump from a farm animal straight to our dinner plates. Knowing that intensive farming can amplify drug-resistant salmonella that jumps to humans, is animal stress management actually the front line of our own public health defense? When we protect domestic animals from overcrowding and stress, aren't we ultimately protecting ourselves? Thank you so much for joining me for this explainer, and I really hope this deep dive gave you a clear, structured understanding of the incredibly complex world of salmonella.